cell is cell. So the goals for today, we're going to talk about what cell theory is. It has three main parts that we're going to go over. We're going to talk about the difference between eukaryotic and prokaryotic cells, so two different types of cells that we see in different types of organisms. The difference, or we're going to talk about some of the primary subcellular components. So you have a cell, and then within a cell you have smaller little what we call organelles that perform very specialized functions to give your cell its overall behavior. We're going to talk about we're going to learn how to relate the structure and the function of these subcellular components. So by looking at the structure of a subcellular component, we can figure out how it's going to behave and what could go wrong with that. So cells, the fundamental unit of life, they're like the basic unit of biology. So everything, all organisms are composed of cells. So they're the basic unit of the organism that gives it its overall properties and behavior. So all cells come from pre-existing cells. So we don't make cells, kind of from scratch, de novo, just like we don't make people from scratch, at least for now. But I mean, in the future, I'm sure this will change. So cells are always going to come from pre-existing cells. So we're going to have one cell, and it's going to grow in size and create multiple copies of all the necessary subcellular components or organelles, and then it's going to split in half. So that's going to be something we're going to learn about a lot more moving forward. So cells, all organisms are composed of cells. They're the basic unit of each organism, and all of the structural and functional characteristics of the organism can be explained all the way down to the cellular level, and that'll give us information on how the organism behaves as a whole. And all cells are going to come from pre-existing cells. So this is what we're going to call cell theory. These are the three parts of cell theory. Any question about any of these? So again, we don't make cells totally from scratch. Your body doesn't say, oh, we need another cell here. Send over all of the proteins and the nucleic acid and the lipids and the carbohydrates and make a cell out of them. One cell is going to grow in size and it's going to split in half to form the second cell. So all of your cells in your body have a lot of features in common with them. They have a lot of the same components and a lot of the same functions, but there's also a very wide array of cell specializations. So just to look at some of these different cell types, so these are red blood cells, these are nerve cells. This is what makes up your brain, gives it its function. Sperm cells, which are useful in reproduction. You have different types of muscle cells. Macrophages, these are a type of cell that's useful in the immune system. So all of these have a lot of properties in common, but they're also very, very different from each other. And the reason they're different is because their subcellular components are going to behave differently. So right now in this chapter, we're going to talk about what's in common for all these cell types. And as we move forward, we'll talk about what's different in these different cell types. Again, the biomolecules that a cell produces are what's going to give it its unique function. So if we have, the red blood cells up here, they're erythrocytes, they're going to be making a lot more hemoglobin than any of these other cells. So based on the genetic structure, the genetic makeup of all these cells, some cell, depending on its environment and its genetic makeup, it's going to produce different biomolecules. And so it can either behave like a red blood cell or it can behave like a nerve cell. But they all have the same exact genetic makeup, all the same nucleic acids inside of them, but they respond to their environment differently to give them very specialized tasks. So again, cells are the basic unit of the organism. So all of these cells are the basic unit, of a person or of any other mammal, but they all have very different roles. So we're going to talk about two different cell types, eukaryotic and prokaryotic. So I like to remember it this way. Eukaryotic sounds like you and me. We're eukaryotic. We have eukaryotic cells in us. Prokaryotic cells, they're a different type of organism that we'll talk about moving forward. So eukaryotic cells are much larger and they have these membrane-bound organelles within them. Prokaryotic cells are much smaller, and they don't have membrane-bound organelles in them. So all of us, are made up of eukaryotic cells, not prokaryotic cells. Prokaryotic cells are much smaller and less complex. So they're going to be usually much less complex organisms. So first we're going to go into prokaryotic cells and some of the structures of those cells, because they're a little smaller and simpler, so it's easier to get a grasp of them. And then if we have time, we're going to get into some of the components of eukaryotic cells.
Can both be single-cell organisms?
It's a good question. So prokaryotic cells definitely can be single-celled or multi-celled. No, I think only single-celled. Eukaryotic, I'm not sure that there are single-celled eukaryotic cells. Usually not. So at least if we look at what the majority of them are, prokaryotic cells are primarily single-celled and eukaryotic are primarily multicellular. I'm sure there are examples of single eukaryotic cells, but nothing's really coming to mind right now. Good question, though. So we're going to talk about some of the components of prokaryotic cells, and by talking about those components, we're going to see how they're similar to eukaryotic cells and how they differ. So prokaryotic cells, they have this cell envelope around them, which is much stiffer than we see in eukaryotic cells. Because since eukaryotic cells are multicellular, they have to send nutrients and information between the two cells, which isn't the case with these prokaryotic cells. They have a much stiffer outer wall on them, basically. The cell envelope is like a stiffer outer wall, and so it maintains the structure of this prokaryotic cell. So this is kind of a cool diagram. This is what a prokaryotic cell actually looks like under a microscope, and this is magnified 32,000 times, which is kind of mind-boggling to me. And so this diagram is nice because it shows you what it looks like in real life, and it gives you this kind of little schematic view of what it looks like in a way that's a little easier for us to understand. So we have this stiffer wall on the outside, and inside of that we have a softer plasma membrane. And this stiff wall is going to give us a structure to the prokaryotic cells, and it's also going to regulate the flow of material in or out of the cell. So cells are really concerned with keeping the environment inside of them very constant. And we kind of know why already, because we know that proteins and other biomolecules are going to behave differently in different pHs, different temperatures, different salt concentrations. Lots of different things can change the function of the biomolecules. So by having this stiff wall on the outside of the prokaryotic cells, it allows those cells to maintain the environment inside of the cell that they need to survive and to thrive. These are some more structures in prokaryotic cells. So we have the cytoplasm. So inside of this prokaryotic cell, we have what we call a semi-fluid. It's kind of a term that you might not see frequently. Semi-fluid is a very viscous and thick fluid, kind of like a syrup. And it's that thick because we have a bunch of biomolecules dissolved into it. This is a cytoplasm, sorry. So the cytoplasm is the semi-fluid inside of the cell. And it's water-based, but it has a ton of biomolecules dissolved into it that make it very thick. And it doesn't, flow like water does. It's more like a syrupy or viscous solution. And so that's what's going to be inside of the prokaryotic cell, and because it has this stiff cell wall and this cell membrane around it, it's going to allow it, no matter what conditions it's in outside of the cell, to keep that semi-fluid just the way it wants it to survive. We have the nucleoid. This is the region where the DNA is found. So you might have seen in previous classes or other pictures that in eukaryotic cells there's a nucleus, which is a little membrane-bound region where the DNA is kept in this very compact arrangement. But prokaryotic cells, they don't have these membrane-bound organelles like eukaryotic cells do. So they just have a region where the DNA is usually found. So we saw in eukaryotic cells, we saw the DNA that was like a long, kind of a twisted ladder like this. And it's just like a long chain like that. In prokaryotic cells, we actually have a plasmid, which is going to be circular DNA. So in prokaryotic cells, the DNA is circular and not a long chain like we see in eukaryotic cells. So the plasmids, or the DNA, is usually found in the nucleoid, which is the region where the DNA is found, and it sits in the cytoplasm, which is a semi-fluid solution inside of the cell. So in prokaryotic cells, we also have these ribosomes. So ribosomes are what's going to translate the DNA into proteins. So basically what a cell is going to do is it floats around in this environment, especially a prokaryotic cell like this. It's going to float around in its environment, and based on the cues it gets from its environment, it's going to use its DNA to guide the production of different proteins. So for instance, if there's a lot of food nearby that needs to be broken down and brought into the cell can use its DNA to create proteins that are going to take that food, break it down, and bring it inside for the cell to use. So this is sort of how cells work. They have their instruction manual in the form of DNA in here. They have the proteins that they make to interact with their environment so they can keep getting enough food and getting rid of their waste to survive. And then they have a cell wall or a cell membrane around it to keep their inner environment very well regulated and tightly controlled. And inside of the cell, we have a fluid called the cytoplasm that's just full of biomolecules that it needs to keep inside of the cell for the cell to survive. Does this sort of make sense, like how a cell would use these biomolecules to do what it needs to do?
What would trigger the DNA to start like producing these proteins? Like I get that it can like quote unquote sense it, but what actually triggers the DNA?
That's a great question. So it's a circular loop. So what happens is the proteins are going to sense the changes in the environment. Because of their structure, they're going to be able to bind to different things. And based on how they bind to different things in the environment, they can send other proteins to the DNA to copy parts of the DNA to send to the ribosomes to make more proteins. Let's think of a better way to explain that. So you have a cell that already has proteins in it, and those proteins interact with the environment. And based on how they interact, they can start like a chain of events that sends more proteins to the DNA to copy specific regions of the DNA to make more proteins that it needs to take advantage of its environment. So it's kind of cool. I mean, these are so small. You know, like we saw, I guess I can't go back when I record. I think that's the problem. So now we have eukaryotic cells. So eukaryotic cells are a lot bigger and more complex, and they have these membrane-bound organelles. So like here, for instance, we have the nucleus, and that's a membrane organelle that contains all of the DNA, just all one tiny confined area. And then we have all these other organelles that we'll talk about moving forward. But the important part about the membrane-bound organelles is that they allow each small region, so each of these small little regions can control its own environment. And based on the environment that it has, it's going to change the function of that organelle. So what happens in the nucleus is very different than what happens in the lysosome. So the lysosome is an organelle that's going to keep its pH very, very low. And this is where we could put organic matter that we consume to break it down. So if you have a really long protein chain that either was floating around the cell and has become damaged, or that you ate and made its way into the cell, you could put that into a lysosome, and the very low pH will break that protein down into its individual building blocks that can then be recycled and used to make other biomolecules. So kind of a good analogy for this is like all of the organelles are sort of like different factories. And just like in real life where each factory has a very different environment, all of the organelles have very different environments, and it makes them a lot more efficient. So if you had a factory that made TVs, it'd be very different than a factory that made cars. And because of those differences in the environment, each factory is a lot more efficient. So just like in cells, each of these organelles has a very different environment inside of it, and that organelle makes it much more, that environment inside the organelle makes it much more efficient at doing its job. Yeah, good.
In these like factories and organelles,
yeah.
Is there anything that doing these things to make it happen, or it just happens automatically?
Yeah, I mean that's the cool thing about biology is it just happens if you have the right mixture of biomolecules in the right environment, they're going to perform all of these functions.
So the cell is just making an environment and putting the elements there so that it does it.
Yeah, and we're just made up of a bunch of cells, and somehow we can do all of these like crazy complex things, which is like really amazing. That's exactly what happens. Is inside of the cell, all that happens is you carefully control each of these environments of the organelles or of the cell as a whole, and you control those environments with biomolecules. And based on the environment, you can produce and use biomolecules differently. And so we have neurons that have different environments inside of them, or blood cells that have different environments, and they all perform very different functions and give us like really complex behavior. Oh, okay. So we'll go over the key ones moving forward. I just want to kind of show. Okay. So we can start going over the organelles, but it is really, I think it's really amazing that biology can do all of this complex behavior with just such a simple mechanisms. And what's really even cooler, I think, is that as sophisticated as we are, we couldn't make anything as complex as a cell. These things that just naturally reproduce inside of us and different animals and plants is like way beyond any technology that we have. Which is why biology is awesome and you should all study it. Okay, so let's start going over a few of the organelles that are in eukaryotic cells. Again, eukaryotic, like you and me, those are what we're going to find in mammals and not in small single-celled organisms. So we have the nucleus. So this is like the nucleus. And inside of the nucleus is the DNA. And again, the point of the DNA is we'll go over this image a lot more in the future, so don't worry about all the details. The point of the DNA is to direct protein production. So that's why we have the DNA. And the DNA in eukaryotic cells is going to be in the nucleus. So the nucleus is like the command center of the cell, which is something I learned for the first time when I was in high school. Like I've literally never forgot that description because it's actually a really like apt description of what the nucleus does. It has all of the instructions for protein production inside of it. And the protein production is what's going to drive all of the cell function. So the nucleus is really like the control center, the command center of the cell. So along the outside of the nucleus, we have the nuclear envelope. So the nuclear envelope is made up of phospholipids, like we saw before. And these phospholipids just form a bilayer based on the way they interact with water. And they keep they maintain the integrity of the nuclear envelope. So they make sure that nothing is just getting in and out just by flowing in and out. Things can only go in and out through the nuclear pores, which are small little holes in the nuclear envelope. So we can send some things, like copies of the DNA, out of the nucleus, but things aren't just like floating away. So it's like tightly controlled, but it also lets some things in and out. So within the nucleus, we have chromatin, and we'll go over this more as we move forward. But chromatin is a mixture of nucleic acids and proteins. So the reason there's proteins in there is because the DNA in all of our cells is like hundreds of meters, maybe kilometers long, but it's all carefully packed into these little cells. All of the cells, we have trillions of cells in our body, all with the same DNA, and the DNA is, maybe a kilometer long, but it's all carefully packed, and it's packed that way because we also use proteins to make sure it's all tightly, organized. It's not like when I put my Christmas lights away and I just like jam them into a box and they're all tangled. It's like carefully spooled around these proteins so it can get nicely organized and packed really densely. So this is the first organelle that we've talked about with eukaryotic cells. It's the nucleus. It contains the DNA. It's like the command center. Now we have the ribosomes. This is where we're going to be making our proteins. This is like the factory of the cell. This takes the instructions from the DNA, which is in the nucleus, and it makes proteins from it. So information comes from the nucleus, through the nuclear pores, to the ribosome, and it makes proteins. So the nucleus is like the command center, ribosomes are like the factory. So we've already seen two different kinds, or really three different kinds of biomolecules so far. we have the nucleic acid that makes up the DNA in the nucleus. We have the proteins that are made of amino acids that are really going to be performing all of the functions for the cell. And we have the phospholipids, which make up the outside of the nucleus to form the nuclear envelope that keeps everything tightly contained, right where it should, right where it should be.
How does it know where to go?
That's a really good question. Okay, the easiest answer. Okay, well, so the most relevant answer to what we're talking about now, it can be put into a vesicle made by the Golgi apparatus and sent around, and that's why we call the Golgi apparatus like the post office, because it moves things around right where they need to go, and it can move along like tracks within the cell to move them to where they need to go. The slightly less intuitive answer is that everything in the cell is just like constantly shaking and moving around. So cells are so small that just the thermal motion from things vibrating around can move things all throughout the cell. And proteins, wherever they need to go, they're going to be binding to what they need to work with. And so it's not so much that they know where to go and they're never in the wrong place. It's that they go to a lot of different places, and when they're in the right place, they're going to be binding to what they need to work with. But that's a really good question. I think kind of the underlying question to like a lot of what I'm hearing is like how do cells know how to do what they need to do, even though like if they're so small and no one's there like directing all the controls. And all of this really just happens spontaneously, just based on the proteins that get constructed from the DNA. If you start from that, those proteins can interact with the environment and they can work with the DNA to change their environment and to change different biomolecules to get what they need to do. And since all cells are coming from pre-existing cells, if you already have that environment, it can just grow and replicate and reproduce. And that's sort of what's amazing about cells is they just copy their DNA, and the cell will divide, and you have two cells that are totally viable on their own, capable of existing in different environments. It's kind of hard to explain. I don't know a better way to explain like how amazing cells are. But if you think of anything, you could help me express it. That would be nice. So I always just think it's like the coolest thing ever.
Are you gonna make a label stuff on the quiz?
You won't have to label it, but you will have to describe what a Golgi apparatus does, or why, like the difference between a smooth ER and a rough ER, like why one is smooth and one is rough.
And one is smooth because they don't have rough.
Exactly. Yeah. So you also, the last part of the endomembrane system is the lysosomes, and this is a small vesicle or a small little organelle that has a very low pH, and it's going to break down other biomolecules and, recycle them to be used at a later time.
Okay, I see you have the word in quotation marks workshop. Is
Yeah.
that endoplasmic
Yeah.
reticulum, or does that refer to the smooth?
No, that's for endoplasmic reticulum.
Okay.
Yeah. All right. So, was everyone ready? Is anyone still copying? Good. Okay. So, one of the organelles that we're going to talk about the most moving forward in eukaryotic cells is the mitochondria. So what the mitochondria does is it takes in glucose that we eat in our food, and it breaks it down into a smaller unit of energy currency, basically. So we take this glucose and we're going to break it down into ATP, into lots of different small molecules of ATP, which we'll talk about, moving forward. So don't worry about exactly what it stands for now. But what happens is glucose contains a lot of energy. So the glucose that we eat in our food has a lot of energy in it. Way too much energy to be spent on like a single cellular action. It'd be like having a $100 bill and like trying to go and buy a pack of gum. The cashier would flip out at you. So instead we break our $100 bill into lots of like $1 bills. So we break our glucose up into lots of ATP molecules. And these ATP molecules can be used to convert biomolecules or to make reactions happen. So I was a little bit incorrect when I said everything happens spontaneously. You need to be constantly ingesting food and oxygen to make everything in the cell happen. But if you're ingesting that food, then your cell can break down the food and make ATP and spend that energy to make different things happen and to keep your cell healthy and alive. So it's sort of like the way a water wheel converts the flow of water into electricity. So it just changes the form of the energy into a way that's more useful for us to use. And it does that by creating a difference in potential energy between the inside and the outside of the organelle. So potential energy is exactly what makes water fall from a high place or anything fall. It's energy that can be used for another task. We'll get more into these later. It's kind of hard to know where to start with things, but what the mitochondria does is it exploits the difference in potential energy to make energy molecules for the cell to use. So we'll just do one more slide and then we'll call it a day for today. So animals don't have chloroplasts, but plants do have chloroplasts. And what the chloroplasts do is they take in light energy from the sun and they make that into glucose that plants or animals can then use to keep the cells healthy and alive. So chloroplasts are kind of like a solar panel. And they are they have this function of the solar panel because of a pigment inside of them called chlorophyll. So chlorophyll is a pigment in the chloroplast that absorbs the sunlight, takes the energy, and uses that energy to make glucose, which can then be sent around the cell. Or, when we eat plants, we take the energy that was taken from the sun and put into glucose, and we eat that glucose, and now we have energy to run around or, to study bio or things like that. All right, any questions? I think this is a good place to stop. No. Okay, so why don't we take a short break, and then if you want to ask questions during the review session, it'll be from like 7:05 to 9:45. I'll just be waiting around.
Can you go a couple slides back?